Hello and welcome. I'm Steven, and this is my computational chemistry presentations on designing OLEDs with DFT calculations. There are two parts in this lab, and before we start, allow me to introduce you the software we're going to use in the first part of this lab. It's in the NanoHab page, and it's called AB Initial Simulations with Orca. We are also going to use VMD later on after we obtain the output log. The Orca allow us to perform AB initial simulations on the molecular system and allow us to do the geometry optimizations with or without coatomic ionization energy calculation. And since we have already obtained the geometry coordinates from our canvas page, we don't need to use other software such as Siesta to obtain the coordinates by ourselves. We simply just have to plug the coordinates into the system, which I will show you later on in this presentation. And the cube file is gonna be used later on in the VMD to do further on analysis in the HOMO and LUMO energy gaps. And now this is what we're gonna work on in this lab. What we have is two cut molecules, and what we want to do is to analyze whether they will be a good HTL or ETL layers. But what is ETL material, HTL material, and OLED? Well, there are two main classes of organic light emitting diodes. OLEDs, so-called small molecule based light emitting diodes, and PLEDs, so-called polymer light emitting diodes. Right, and a typical double heterostructure small molecule OLED consists of three organic layers sandwiched, just like this. What we got is positive charged electrodes at the top, the negative charged electrode on substrate at the bottom, and we've got ETL, EML, and HTL. And the full name of the three layers are the electron transport layer, the ETL, the hole transport layer, HTL, and at last the emissive layer, which is the most unique one because it consists of a light emitting dyes dispersed in a suitable host materials, and it's often same as the HTL or ETL material. And what we can do from here is to analyze this by plotting the geometry coordinates of the optimized molecules and also recording the energy given. And last, we are able to use the output log to calculate and do further analysis on the HOMO and LUMO energy, which is going to be the main task of this first part of the lab. And now we're ready to begin. I did try to use my laptop screen recording software to film this process to make our life easier, but it's not really working as I expected. So what I'm gonna do is use the old school way to use the screenshot and talk you through it. Hopefully you understand. This is the first impression of the AB initial simulations Okra when we first open it. We have to change several things. What we have to do first is to click on this thing to change our task. We have 4 tasks given, and what we're gonna do today is the geometry optimization, as we would probably expect it. And we also need to change the advanced options from no to on to allow us to do to change several settings later on, and also I have changed the molecule to H2O and to make our life easier. 3 more different settings is going to pop out once we've changed the advanced options from no to yes. We first need to change the coordinates from Z matrix into Cartesian to allow us to put in the right coordinates as provided, and then we have to plug in our coordinates. If we right-click the blind space in the atomic structure, we can get several index like this, and we press upload. There is going to be an upload page on your laptop, hopefully, and you simply just chuck in or plug in the file you're going to be analysis on, and it will upload the geometry coordinates into the atomic structure page straight away. And what we have to do further on is to delete this number so the system is going to work properly. And I think I have to mention the spin and charge on this section specifically, because when we are doing the cation, anion, triplets, triplets, singlets, different type of stuff, we have to change the spin and charge when we have to. So the spin has to be changed in the cation and anion sides from +1 to -1, and the charge has to be changed from ×1 or ×2 or ×3 in order to make our calculation valid. And now we would have our final settings, the energy expression. We can see there are 5 several methods we can use, but what we're going to use today is the density functional theory. And this is what our settings should be look like for this lab. The XC potential is going to be B3 LYP, and the basis set is going to be 6-31G. Notice that the geometry reduction bottom has to be open to allow the experiment to work properly. And after this, we simply just press simulation, and the computer is going to do our job for us. There is going to be different results we could possibly see. Simply just press this button here, and you get what you see. And for now, if I want to do the calculation of the ionization energy, the electron affinity, and also the HOMO-LUMO energy gaps, we're going to use the output log to help us. So after we run all 8 different simulations, you're going to download the output log, press this button, and this is what the output log is going to be look like after you download it. You can notice that there is going to be a lot of data from the index here. So what I did is use the search tool to search the data I want and make a form out of it. And here's the result table I made after I run all the calculations. This is the settings I used, and this is all the data, and the unit is kilocalories per mole, same as the electron affinity and ionization energy. I have changed the answer to 2 decimal places for reading purposes, and if we consider the stability simply in terms of energy, then a triplet state is definitely more stable than a singlet state because of the lower energy. The lower the energy, the more stable it is. For example, a triplet state is of lower energy than the singlet excited states. Before we move on to the HOMO and LUMO energy, we first need to understand the concept of it. HOMO stands for the highest occupied molecular orbital, while LUMO stands for the lowest unoccupied molecular orbital. The LUMO is higher in energy than the HOMO. Of the orbitals that have electrons, the HOMO is the highest in energy, and of the orbitals that do not, the LUMO has the lowest in energy. This means that they are closest in energy out of all orbitals in the molecule. Due to the energies of these orbitals being the closest of any orbitals of different energy levels, the HOMO-LUMO gap is where the most likely excitations can occur. Hence, it is the most important energy gap to be considered in this experiment. And here are some simple facts of a HOMO and the LUMO system. The higher the aromatic system is, the smaller the HOMO-LUMO gap is. The greater the mobility of pi electrons in large conjugated pi orbital systems, the greater the distribution of the energy throughout the molecule, stabilizing it. Hence, the smaller HOMO-LUMO gaps correspond to the better stability. And now let's do the HOMO and LUMO gaps. We first need to open one of the output log saved in our laptop and get a notebook version like this. We press Ctrl F to the data we needed, in this case the orbital energies, and we can get a list of number with showing this. OCC applies to the occupation and EEV and applies to the energy. So we scroll down to the energy gaps, the HOMO and LUMO energy gaps, and we get those data for further calculations. And after the precise calculation, we get the lowest HOMO energy goes to the ETL, the lowest LUMO energy goes to the HTL, both fact. And the PA, however, has the highest LUMO energy, while the PB has the lowest HOMO energy. In this case, we can draw the conclusions that PB is the ETL and PA goes to the HTL. While the higher energy HOMO allow the positive Hall transfer to EML, while the higher energy LUMO prevents the negative charge transfer to HTL after transition to EML, and the PB goes to the other way. And for the last part of part 1, I've used VMD to plot the HOMO and LUMO graphs for PY and for PYPB or PYPA. And here are my results. The setups are quite easy as well. Just simply follow the instructions, and you create two rep, and the ISO value would be positive 0.05, and the other one would be negative 0.05. You change the drawing method to ISO surface, and you show the solid surface in the final window. And each graph suggests different types of molecules. So in this case, index number 3 suggests the PA LUMO diagram, and index number 4 suggests that it's gonna be the PA HOMO diagram. Thank you. This will be the end of part 1 of this lab. So in this lab, we have used the AB initial simulation system in NanoHab to run the simulations of the geometry using the geometry coordinates of 2 molecules. We have used the output log of the simulation to do the analysis and calculations of electron affinity and ionization energy. We have also used the data to understand the ETL, EML, and HTL system. And at the end, we have used the VMD system to see the actual image of the molecules and understand the HOMO and the LUMO energy gaps. For the second part of this lab, what is shown on the page at the moment are the steered molecular dynamics simulation, and it allows the simulation of a molecule once submitted to a virtual traction along its axis, and a group of atoms is kept focused on one side of the molecule, and the force is applied through a virtual spring. And now let's move on to the application of the software. As you can see, we need to upload files before doing anything. And when you click on that, there's 3 files you need to upload: the initial geometry PDB file, the structure file, and the fixed atoms file. If you go onto the canvas page, there are 5 files provided for us for this lab. We have 1 meter PDF, PDB, a 1 meter PSF, SMD PDB, parameter, and fixed PDB. The 1 meter PSF is a PDF file. It's also called a protein structure file. It contains all the molecule specific information needed to apply a particular force field to a molecular system, and it's obviously a structure file. The initial geometry PDB file is the 1 meter PDB file. It contains the initial geometry of the protein before performing the SMD simulation. And the fixed atom file obviously is the fixed PDB file. And before we move on, we have to click this button here to change the no fixed atoms from no to yes. And now let's move on to the next page. So we need to upload the remaining 2 files, the parameter and the SMD files, into 2 different settings. After we've done that, we have to change some settings. So we have to remain the temperature to 300 and change the number of bins into 10,000. The number of averaging bins and DCD frequency remains like it was. And for this one, this is the most important step. We have to change the velocity from whatever that is to 0.05 to save us some time, or it's gonna take at least 4 hours, I think, to get the final data. And we can change the direction of X, Y, Z, whatever we like. So I have choose for this one to change the direction of X to 1, and Y to 0, and Z to 0. And the force constant remain as it was. And now it goes on to the last step. We run the simulation and get a plot diagram like this, the energy plot diagram like this, but it's not really what we want. What we want is the VMD movie files, zip files, right here. And after that, we get a list of data, and we press this small button here to download the zip file using the VMD movie. So it's kind of an application from SMD to VMD movie files. And after we open that zip file, we get 2 small files in that folder. We get PSF and DCD files. So we open the VMD software like we did for last time, and we upload the PSF file first. We may not get anything out of it, so what we have to do is right click this bit here and choose loaded with loaded data into the molecule, and we loaded list data. And after that step, we can get a clear image of our protein structure stretching out. So because it's actually a photo, you can actually see the floating of this protein. So basically, it's something like this. So it's all crowded and stretching like this. It wasn't that clear in terms of structure, but you can still see some chemical bound in. You can see some water molecule floating around, and you can actually see some chain, like a polypeptide chain, for example. So in order to show the sequence in a clear sight, you press the representation button, and you can create a wrap. So for the new wrap, I changed my drawing method into new cartoon, and change my coloring method into secondary structure. And after that, you may see a clear image. And now we got it. So we have a much clearer image compared to before. And the settings I use for the first time is 5,000 steps, and velocity is 0.05. That's my settings. This actually allows us to see a much clearer image, and we can actually see the water molecules nearby. Speaking of water molecules, the chemical formula of molecule is H2O, and hydrogen bonds form between neighboring hydrogen and oxygen atoms to adjacent water molecules. The attraction between the individual molecules creates a bond known as the hydrogen bond, which I consider is essential in this lab, the hydrogen bond. So the peptide bond supports the conclusion that hydrogen bonds stabilize the folded form of proteins, and with water molecules started to conjugate and stabilize, as we may notice from the diagram, I'm thinking the protein is losing hydrogen bound molecules, and due to the lack of the hydrogen bond, the protein structures started to concentrate together, and I think that's where the structures begins. Another fact of protein and hydrogen bonds are hydrogen bonds provides the most directional interactions that underpin protein folding, protein structures, and molecular recognition. And the core of the most protein structure is composed of the secondary structures such as the alpha helix and beta sheet. And if we bring back to the original diagram of our lab, since our aim of this lab is to investigate a single alpha helical segment of the 2B part of the Wymanton intermediate filament dimer, which is labeled here, and we understand it's a protein structure, secondary protein, we got a helix going on, and after analyzing with the BMD diagram, we understand the water molecules move and how this is going to affect the hydrogen bound in the protein. And with that in mind and doing all those VMD analysis and output log calculation, we have built further knowledge onto the protein structure, water molecules, hydrophobic and hydrophilic nature of the molecule, and also the structure of protein, especially the secondary structure. Thank you for your time, and this will be the end of my presentations. I realize it's longer than it should be, but I just want to make sure I have covered everything and answered all the questions from the exercise. Thank you again, and I hope you have a nice day and week. Thank you.